Assalamu alaikum wa rahmatullah. So let's go over the last part of the transcription regulation lecture. So in this lecture, I'll go slow so that you can understand the concepts that I'll be talking about. So here what we will be doing is that I'm gonna explain a couple of techniques that we use to measure the levels of RNA. And we also use another technique not only to see the measure of to measure the level of RNA, but also to see where it is expressed exactly. Okay, so the idea here is that the question is, why do we want to know the level of RNA? Because that tells us something about how active the gene is in terms of expression. Okay, so just like the lac operant, for example, we know that in the presence of lactose and presence of glucose, there is little expression of the lac operant. Okay, however, when glucose is removed, expression of the lac operant increases. Okay, so the amount of RNA that is present in bacterial cells is very high. Okay, so that tells us something about how active the gene is. So let's start with the first technique. So it's known as a northern blotting, okay, or northern blot. Now the idea is that when we talk about southern blot, this technique was invented by a scientist known as Southern. Okay, so other scientists in Chicago came up with this technique. It's the same idea as southern blotting, and they called it northern blotting. Now the idea is that they really wanted to be funny when they came up with this name, and they said, southern blotting, so we are from Chicago, we are from north of the US, so we will call it northern blotting. Anyhow, scientists, geeky scientists, try sometimes to be funny. So the idea here is that northern blotting, we use it to identify how much, relatively, how much RNA there is in a biological sample, like bacterial cells, for example, or human cells, and so on. So here's the idea, okay? So the idea is that we take a sample, we like cells, okay, for example, and we open up this sample, releasing all of the RNA molecules. We take the RNA molecules. Now someone might say, well, how about the DNA, how about the protein, and so on? There are certain ways that we use to isolate RNA molecules. So we take the RNA molecules and we separate them in a gel, just like the agarose gel that we talked about for DNA, same idea. So we take this, we take the RNA, all of the RNA molecules, and we separate them in a gel. Okay, so here the idea now is that in this gel right here, we have these are the wells, and the sample runs from again the negative end to the positive end, same idea. Here we have the large RNA molecules, and in the bottom we have the small RNA molecules. They migrate really fast, these small RNA molecules, relative to the large RNA molecules. Now notice here in this gel, it's smeary. Okay, so you don't really see distinguish or distinct RNA bands, because we have a lot of RNA molecules of different sizes, so there are no distinct bands, right? Rather it's smeary, mamsuh. Okay, anyhow, so we have RNA molecules of different sizes. Okay, so here we have sample 1. Okay, this is sample 1, sample 2, sample 3, sample 4. Okay, then we take it and we transfer it to a membrane. Okay, this is the name of the membrane, a filter membrane, a piece of paper it stands right here. Here we have the gel, and the RNA molecules move upward. Okay, why? Because there is a buffer right here, or there's a solution that is absorbed by a piece of paper, this piece of paper, and it carries the RNA molecules upward to the membrane. So the same idea here, we have this membrane right here having like a replica, a mirror image of the gel. So if we have large RNA molecules here and small RNA molecules right here, we will have the same exact distribution of RNA molecules on the membrane or that piece of paper. So we take it and we add a probe. It's a DNA probe, really. It can be RNA, but we use a DNA probe. So it's the same thing. And this DNA probe is actually radioactively labeled or fluorescently labeled, whatever. So it's labeled in a certain way, and we add it to the membrane. Now, if there is a messenger RNA that is complementary to the probe, this messenger RNA would hybridize to this probe. Okay, so we don't really have to know the whole sequence of the RNA, we only need to know part of it. Okay, now, so right here we have a probe that actually hybridizes to the messenger RNA. Now someone might say, whoa, wait a second, so you can have a hybrid, okay, you can have one strand RNA, one strand DNA, and the thing is, yes, of course we can. Why not? You can have that. Actually, we do have it in the cell, right? We do have a hybrid of RNA, DNA. Remember when we talked about transcription, and when the RNA polymerase transcribes DNA into RNA, and I said that part of the messenger RNA hybridizes with the DNA, right? So you do have this RNA DNA hybrid. So you have the probe now hybridizing to the messenger RNA. Okay, someone might say, well, wait a second, you have a lot of probes in here, so you do have a lot of signal. This is right, but all of these probes that are not bound to the messenger RNA, they are washed away, they are removed. So at the end you would have only the probe that hybridizes to the messenger RNA. Okay, so you have something like this. These are the results after we wash away the unbound probe and we expose the membrane to the X-ray film, and we see something that looks like this. Okay, so remember we have sample 1 here, sample 2, sample 3, sample 4. And you do have bands in sample 1, sample 2, and sample 3. Now think about it here. What are the information that we can get from this? Well, think about it. We know from this image that samples 1, 2, and 4 have bands that hybridize to the probe. Okay? So what do we know? We know that, but not sample 3. So we know that samples 1, 2, and 4 express the gene that hybridizes to this probe, right? So because now we know there's mRNA and it binds to the probe. We know what the sequence of the probe is. So we know that it hybridizes to this messenger RNA that exists only in these samples 1, 2, and 4. But sample 3, there is no signal, there is no band whatsoever. So what does that mean? That in this sample there is no messenger RNA, meaning that there is no expression of that particular gene. Great. So that's the first piece of information, that the gene is expressed in these samples. Now there's something else that we know. And the other thing that we know is that the size of the messenger RNA, we know what it is. because if that's the top of the membrane, it means this is the top of the gel, right? So the messenger RNA molecules that are large exist right here, and the small RNA molecules exist in this region right here. So we know what the sequence of the messenger RNA is in here. Great. So that's two. There is another piece of information that we can learn from northern blotting, and that is how much RNA is there in the sample, or how much the gene is expressed. Now, I tried not to emphasize a lot in southern blotting about the thickness of the band, and I said don't worry about the thickness of the band, okay? But here it's important. Why? Because the more intense the band is, the thicker the band, the more the gene is expressed. So right here we have a probe for a certain gene, certain messenger RNA, and we have samples from different parts of the body. So we have right here, you can read them: cerebellum, مخيخ, we have liver, we have spleen, we have thymus, lung, and so on. So we have mRNA molecules taken from different parts of the body. We separate them through the agarose gel. Again, we transfer to a membrane, we add a probe for that specific messenger RNA, and notice here that we have a band that is not really that thick compared to this band or that band. So we know that this gene is highly expressed in the cerebellum compared to, for example, total brain. You know, if you take total brain, or if you take the retina, or if you compare them to the pancreas right here, okay? But the gene is not, there is no band whatsoever in these tissue samples. There is no band in the lung specimen, the lung sample, or the thymus, or the spleen. What does that mean? It means that the gene is not really expressed in these tissues. Okay, so we know number of information. We know what the size of the messenger RNA is, which, by the way, it represents the exons, not the introns. We don't see the pre-mRNA. Why is it that we don't see it? Because processing of messenger RNA is very fast. Okay, so we do not see pre-messenger RNA. So we know what the size is, we know where it is expressed in what samples, we know what the size is, and we can tell how much of this gene is expressed based on the thickness of the messenger RNA band. Okay, so this is really northern blotting. Now, this course is about thinking. Okay, so now let's look here. We have three samples, 1, 2, and 3. We have total messenger RNA separated, molecules separated through the agarose gel again. We add a probe, and here what do we see? We see a band that is at a certain size, whatever the size is. Let's say it's about kilo base long. Remember, it's not base pair because it's single-stranded. It's messenger RNA, so it's 4 kilo base. Okay, that's in sample 1. Look at sample 2. Oh, there are two bands in here. One that is about 4 kilo base long, and the other one is about, let's say, I don't know, it's just, you know, arbitrary number. It's like 2 kilo base messenger RNA. So there are two bands. Now, look at sample 3. There's only one band right here, and its size is about 2 kilo bases. Now, think about it here. Why is it that sample 1 has one band, 4 kilo base long, sample 2 has two bands, 4 and 2, and sample 3 has only one band, which is about 2 kilo base long? Think about the reasons why we have different length or different sizes of messenger RNA. One, interpretation. You can have a probe that is not specific. Okay, so it binds to one messenger RNA in one sample, but it binds to two messenger RNA molecules in two samples. So this could be the gene of interest that is expressed, and this is an artifact. This is false because of non-specific hybridization. Okay. Probably sample 3 does not express the gene of interest. We expect its size, the size of messenger RNA, to be about 4, but it expresses the gene that is really non-specific. This is not the one that we are interested in. So it could be that the probe is not specific. That's one. Number 2, second interpretation. You can have in these different 3 samples, let's say from different tissues, brain, liver, lung, you can have the gene expressed, the same gene expressed in all of these samples, but it is processed differently. So you can have alternative splicing. You have an exon that is extra in sample 1 for the gene. In sample 1, you can have the gene producing messenger RNA that is processed differently in 2 different ways, producing 2 different messenger RNA molecules. Okay, having an extra exon, this is lacking the exon. And in sample 3, you have the splicing of the messenger RNA, so you have only the low molecular weight messenger RNA, but not the large molecular weight messenger RNA. Okay, so you can have alternative splicing. You can have expression of isoforms, the different genes. Okay, but they do share the similar sequences of the probe, like let's say histones. I mean, look at what is the difference between histone 2A and 2B. Hardly anything. Okay, there are differences, but they are homologous. That is, they are highly similar in sequence in terms of DNA or even messenger RNA. So if we use a probe for H2A, it can also hybridize to the probe for H2B. Why? Because they have highly similar sequences. So the same probe may bind to 2 different genes. Okay, so there are many interpretations for this gene, for northern black. Okay, great. Maybe there are different interpretations, by the way, as well, for why we have different sizes of messenger RNA. So in the exam, if I give you something like this, you really have to think. I mean, this molecular biology course is not about memorization. You have to link information together. So let's look here. Now, what do we see in here? We have 3 samples. 1, 2, 3. This is the molecular weight marker. Okay, this, we know what the sizes of RNA molecules in this sample, and we can compare them to the messenger RNA that exists in these samples. So right here we have sample, let's see, I'm sorry, okay, so let's look here. So we have sample 1, sample 2, sample 3. So what do we see? That's the gene of interest. We see the band getting thicker in sample 2 versus sample 1, and in sample 3 versus samples 2 and 1. Great. What does that mean? It means that the messenger, there's a lot more messenger RNA in sample 3 than sample 2, and in sample 2 than sample 1. This means that the gene is expressed more in sample 3 than sample 2, and more in sample 2 than sample 1. When do we see such a thing? Well, let's see. Let's take the lac operon as an example. Right here we have a sample with little lactose and a lot of glucose. So we do have very little expression of the lac operon. Look at sample 2. Here we reduce the level of glucose, right? And we have a lot of lactose. This means that there should be more expression of the lac operon, and that's why you have more of the mRNA of the that is produced from the lac operon, the polycistronic messenger RNA. In sample 3, we have no glucose whatsoever, and we have a lot of lactose. So there should be a lot of expression of the lac operon, and that's why the band is very thick, right? Well, someone might say, well, wait. I think you're fooling me. أنت عمالك تخدعني هون. I say, why? He said, well, wait a second. Maybe what you did in here is that when you added total messenger RNA of the sample, you added, let's say, 1 microgram of messenger RNA in here. Total messenger RNA. And in sample 2, you actually added 10 micrograms of total messenger RNA. So relatively, there should be more of the mRNA of the lac operon, right? Because I'm adding more total messenger RNA. And in sample 3, you added 20 micrograms of total RNA, and that's why you see that the band is even thicker. So what do you see here? You see little mRNA from the lac operon. You have a lot more in here and a lot more in here. Why? Because you're adding more sample in number 3, right? I mean, think about it. So someone might say, well, yeah, you're fooling, you're cheating in here. So the increase you see in here is really artificial. It's false. It's not right. Now I would say no. My experiment is right, and the reason is that I'm using here a probe for a housekeeping gene. What is a housekeeping gene? A housekeeping gene is a gene that is always expressed, and it doesn't get affected. It is not affected by whatever condition the cell is placed at. Okay, so for example, the expression of the lac operon changes because the environment is different. Here we have a lot of lactose and there is no glucose, right? So the expression of the lac operon changes according to the environment, to the surroundings of the cells. But a housekeeping gene like histones, for example, should not change that much. Okay, why? Because it's not affected by whether there is glucose or lactose. Or we can take another gene like, for example, in human cells, actin. Actin is important for maintaining the shape of the cell. So the level of actin should not really change that much. Same thing with tubulin. Expression should not really change that much. So if you look at the expression of tubulin or actin or any other housekeeping gene, you see that the thickness of the band is what? Look at it. It's the same, right? So what does that tell you? That really I'm not adding different amounts of total RNA in these samples. Okay, that's the idea right here. Okay, so that's a proof that the total amount of sample that I'm using in these samples, or that I'm loading, okay, in these wells, really is the same for all these samples. So the increase that I see in here is real. So this is a ruler that I use to indicate, to prove that the amount of total messenger RNA is really the same in all these 3 samples, and the increase that I see in here is a true increase. Okay, so that's the idea of a housekeeping gene. It's a ruler. مسطرة زي the weight that we use to measure the, let's say, the weight of vegetables or fruits, okay, in the vegetable stores. Same thing. Okay, so we use a ruler, we use something as a standard, and this is our standard right here. Okay, so right here, for example, same thing, but what do we see? We see an increase in the amount, on the thickness of the messenger RNA, and that tells me what? That there is more messenger RNA in sample 3 versus sample 2, in sample 2 versus number 1. Okay? Now, is this increase true? Is it real? Well, look at the housekeeping gene. What do we see in here? There is even more of the messenger RNA of the housekeeping gene versus number 2, and more in number 2 than number 1. So that tells me what? It tells me that the loading, the amount of messenger RNA, total messenger RNA that I'm using here is not really equal in these samples. So I'm using more total RNA for sample 3 than sample 1, and sample 2 than sample 2, and sample 2 than sample 1. Okay, and that is so the increase that I see in here is really an artifact. It's not real. Okay, so that's the idea of a housekeeping gene. Keep that in mind. Okay, so this is northern blotting. Now there is another technique, and it's known as in situ hybridization. Remember, we talked about another technique that has a similar name, and it's known as fluorescent in situ hybridization, and we call it FISH. Remember this name because you will hear this name a lot in medicine. It's one of those, FISH is one of those techniques that are important in genetics and in diagnosis of diseases. So this technique is known as in situ hybridization. We know what hybridization means. So again, what does in situ mean? It means in place. In situ means in position, in place. So here in this technique, we are looking at expression, not only how much RNA there is relatively, nisbiyan, we know where this expression takes place. So the idea here is that if I take you learned in histology that if you look at a tissue, there are a lot of cells, right? Different types of cells. So there are epithelial cells, and you have fibroblasts, and you can have inflammatory cells like immune cells like macrophages, for example. And if you look even at and you have endothelial cells that make up the blood vessels. Okay, and you have different types of epithelial cells, right? You have the stem cells, you have the less differentiated cells, and you have the really highly differentiated cells, right? Like in skin, for example. Now, if you have something like a cancer, for example, you have a tissue with a tumor tissue, you would have normal cells, different types of normal cells, and you have different types of cancer cells, right? You can have the benign cells or the primary tumor cells, and you have the advanced aggressive cancer cells. So there are different types. So if you take all of the messenger RNA molecules in this tissue, and you see that there is an increase in the of the messenger RNA in a tumor tissue, for example, you wouldn't really know, you cannot really say that tumor cells express more of this gene than normal cells, because you don't know where this gene is expressed exactly. Is it expressed more in normal cells, or more in epithelial cells, or more in cancer cells, more in inflammatory cells, for example? You don't know that. So the idea here is that we want to know not only how much messenger RNA is expressed in a tissue section, we want to know where it is expressed. And that is the purpose of in situ hybridization. So here the idea is that we take a tissue sample right here, zoom in here. Okay, there are different types of cells. Okay, so what you do is that you take a probe, and it's labeled. Okay, a probe that is complementary to messenger RNA. That's the messenger RNA right here. So this probe would hybridize to this messenger RNA, right? And you add it to whole tissue section, to all of this. So you zoom in here, you look at these cells, they express a this messenger RNA expressing this gene, and you have hybridization happening in this cell or these cells right here, okay? But you don't see any signal coming from these differentiated cells. You only see them in these less differentiated cells. You don't see any signal in these cells right here, in the connective tissue, in the fibroblast cells. Okay, you only see them in these cells, for example. So what can you tell? Well, if there's a signal that is coming out from this part of cells, it means that these cells express this gene, but not these cells, not these cells right here. And depending on the intensity of the signal, you know as well how much of this gene is expressed in these cells. Okay, so that's the idea. So right here, what you see in here is that these are actually Drosophila, embryos of Drosophila. What is Drosophila? It's the fruit fly, ذبابة الفاكهة. We use it a lot to understand our system. So this is labeling of different genes right here. This is the embryo of Drosophila, ذبابة الفاكهة, fruit fly. And you see that there's a lot of signal coming from this region when we probe for a certain gene, okay, and little expression coming from this end of the Drosophila. Look at this expression right here. You see it in the perimeter, okay, بالمحيط of the embryo, and you see some expression right here, okay. Why is it that you see this differential? Why is it that differential expression? Why is it that you see this pattern of expression? Because that's important for development. This would become the head, and the gene specifies that this is the head, this is the tail, okay? Polarity. It's the same idea of having in our system left and right, symmetry. We have top and bottom, we have head and legs, okay, like in animals, for example, they have head and tail, okay, and so on. So you have differential expression in different cells because you have differentiation, okay. So you see that this gene, that's a different gene right here, it is expressed in these cells right here specifically, okay. So that's really important. You see expression of this gene, it occurs in this pattern and so on. So every particular gene has a certain specific expression. Now, if we take this whole embryo and we look at expression of the messenger RNA by northern blotting, we really wouldn't be able to know where this gene is expressed. But by doing in situ hybridization, not only that we see relatively how much of this gene is expressed, we know exactly also what cells express this particular gene. This is an excellent example right here. This is in situ hybridization. And this here is another technique known as immunohistochemistry. I'm sure you learned of immunohistochemistry in histology, but just a reminder, it's a technique that we use to look where a protein is expressed in a tissue section. So we have a tissue section right here, and we add an antibody, الجسم المضاد, an antibody that is specific for a certain protein, and then we stain, we color where this antibody exists. So notice that the color exists right here, okay. So but with in situ hybridization, you see the messenger RNA is right here. The messenger RNA is actually expressed intracellularly. Let's zoom in here. So you see that these cells express the messenger RNA of this particular gene, whatever the name is, okay? So you see that these cells express the messenger RNA right here, okay? So there is no expression in here whatsoever, right? You see some expression in these cells as well. Okay, so let's look at the protein. If you look at it, actually, there is no expression inside the cells whatsoever. Where does the protein exist? In the basement membrane. Right here, that's the basement membrane. So what does that mean? There's no staining inside the cells, no staining intracellularly. Look at here. Look, there is only expression or staining in the basement membrane. So what does that mean? It means that the messenger RNA is expressed inside cells, and these cells then secrete, they release this protein, and it gets localized, it gets embedded, it gets stored in the basement membrane. Okay, so if we only look at the protein, we would say, whoa, wait a second, where does this protein come from, right? And by doing in situ hybridization, we know that these are the cells that are responsible for transcribing this gene and translating it into a protein, and then the cells release the protein into the basement membrane. Okay, so it's not these differentiated cells, okay, rather it's these cells in the bottom that are responsible for this gene expression. So this is in situ hybridization. So the idea again, RNA and protein molecules do not coexist and are present in different places. The messenger RNA exists inside cells along the basement membrane, and protein exists outside the cells in the basement membrane. But so with in situ hybridization, we know what the source of the protein is by looking at the messenger RNA. So we talked about two techniques in here. The first technique is northern blotting. We use it to learn about the level of the expression of a certain gene. We know what if a gene is expressed in a sample or not. We know how much of it is expressed, okay, and we know what the size of the messenger RNA is as well. Okay. So it gives us a lot of information. But with the in situ hybridization, what do we know? We know in a tissue section where a particular gene is expressed. Okay. So these are two techniques that are widely used in molecular biology, and pay attention to the details. Allah ma'akum.